Okay, we'll get going. I want to give Dr. Snyder plenty of time. So it's my great pleasure to introduce Dr. Michael Snyder. So I first met Mike when he was the chair of the MCDB department at Yale, and my first encounter with him was when I actually showed up to interview, and he was talking to all the grad students. He said, "We'll accept many of you, but not all of you." So he put the fear of God in me, right? But he was a great department chair. I actually invited him to be on my committee, and he was always really outstanding. But I want to tell you a little bit about what Mike has done. So Mike grew up in Pennsylvania, actually got a Bausch and Lomb scholarship to attend the University of Rochester, and studied both chemistry and biology there. And after graduating, worked as a technician for a little while, and then went to the West Coast, and this was probably your first experience on the West Coast, right?
Yeah.
Went to the West Coast to do his PhD at Caltech. And so while he was at Caltech, he worked with Norman Davidson, one of these, obviously if he's at Caltech, he's a big guy, right? But Mike did some really cool stuff. He worked in Drosophila and studied the cuticle proteins, and actually found some really cool stuff going on there. He found that clusters of related proteins that are related would cluster together, and he also found that transposons could jump into promoters and mess stuff up, and discovered pseudogenes, I think one of the first examples of pseudogenes in eukaryotes. So he really did some really cool fundamental stuff that was kind of foreshadowing what he would do with genomics later on. After his successful Caltech PhD, where he's published multiple cell papers and PNAS papers, he went to do his postdoc with Ron Davis at Stanford. And so if you haven't heard of Ron Davis, then you don't really know too much. But Ron Davis does amazing stuff, okay? So not to offend you other friends, but yeah, I think you know that already. Okay, but while he was with Ron, he actually developed a method to have expression libraries that he could screen using antibodies, so he could actually have them express the proteins, figure out what it was, and he kind of pioneered that and did some great stuff. And then he went on to Yale. He started at Yale in 1986, and it was initially the Department of Biology, and then it split later on, and he became the chair of molecular, cellular, and developmental biology. And while he was at Yale, did some incredible stuff. When I was there, I remember his lab was always doing proteomic stuff. So they actually did the first proteomics chip. They actually invented RNA-seq, I think, with Pat Brown. Was he involved with that?
Not that one.
Not
but
that much. So he invented RNA-seq, okay. He actually invented paired-end sequencing also. And I think his group was the first group to use high-throughput next-generation sequencing to actually sequence an organism, a parasitic microbe, I believe. Yeah. So you can see that he was just inventing new stuff, doing new applications. So I went from Yale, and I went over to Stanford. And a couple of weeks before I was leaving Stanford, I see Mike show up, okay? And so I accused him of following me. And I said, if you show up at BYU, I know you're following me. Because what happened is Stanford, their chair of genetics department, left to work in industry. And so when you're interviewing to replace a chair, you just have them come out and give talks, right? Because they don't necessarily want their institutions to know that they're thinking about leaving. And so they were having all these amazing high-profile guys come out, and Mike was one of them. And so Mike, interviewed. He decided he wanted to come back to Stanford. Who can blame him? Weather's much nicer than New Haven, right? And came to Stanford to become the department chair just at the time I was leaving. And since he's been at Stanford, he's done some more amazing stuff. So he's been one of the founding PIs on the ENCODE project, about the Encyclopedia of DNA Elements. But he's really gotten into personal omics profiling, and that's one of the topics he's going to talk about today. But his lab is just incredible. I added it up the other day, and he actually didn't know this, but he's received over 150 NIH grants, bringing in, a lot of money. I won't name numbers because you'll feel sad. Okay, but he has over 500 papers. He's invented technologies and other things, and his papers have been cited thousands and thousands of times. He gets close to 10,000 citations a year. Mike is just one of the real powerhouses in genomics and genetics, but at the same time, one of the nicest guys you'll meet also. And in fact, he's so humble, I'll just tell one little story about him. So just before I left Yale, I was participating in the symposium that was set up by our department. I don't know if Mike remembers this. And I did one bad thing. I borrowed his USB drive, which was very early on. No one else had them. And to get my talk onto the computer, I actually erased everything on his drive. So I'm surprised he still doesn't hate me, okay? But the other thing is, after the symposium, we'd had lots of external speakers. And I went up to Mike and I pointed out, hey, all these invited speakers are HHMI. And Mike's reaction was, oh, that's unfortunate. Not saying that they were bad people, but he didn't want the students and others participating to think you had to be Howard Hughes Medical Institute to be successful. And that just really kind of typified Mike to me, that he does amazing stuff, is actually the chair of U.S. News and World Report's #1 ranked genomics, genetics, and bioinformatics schools in the nation. But at the same time, he likes to see people succeed, and he doesn't judge other people by the number, the HHMI or other things like that. So please join in with me to welcome Mike Snyder.
Thank you. Well, thanks for that incredibly generous introduction. I don't think I've ever had anything quite so warm. I will tell you, and I don't remember the symposium or my erased flash drive, but okay. Anyway, I'm sure it all worked out fine. I am going to talk about big data and how we use it to manage health. It is one area that we are very intensely interested in these days. So I like to present this slide because it does frame at least the way we think about human health. Okay, that is to say, we think, I was trained as a yeast geneticist, and I like to think about things in states. So we like to think that most of the time you're in a healthy state, but there are times when you get ill, cold, what have you, or sometimes serious illnesses, and those would obviously be disease states. And I would argue a major goal of us is to try and understand what it means to be in a healthy state, what happens when you transition to a disease, and ideally drive yourself back to a healthy state. Now we know this is influenced by many factors. It's influenced by your DNA, your genome. I'm chair of genetics, but I'll also be the first to admit that it's affected by many other things that you're exposed to, the so-called exposome, pathogens, environmental exposures, food you eat, possible life stresses, lifestyle such as exercise, all influence your health. And I'm a believer that we will be able to understand this in quantitative terms in the not so distant future. People will be born with their genomes already sequenced, and then we'd like to understand what kinds of variants and what kind of context would lead to what kinds of health outcomes. And just as an example of this, if you're at high risk for Parkinson's, you probably don't want to be a pesticide worker because that dramatically increases your chances of getting Parkinson's disease. Now, you may not want to be a pesticide worker anyway, that's a whole different story, but you especially don't want to be one if you're at high risk for Parkinson's. So anyway, these are the things we'd like to do, and again, I do think we can understand this in quantitative terms. So I think many of you appreciate there's been a revolution in technologies over the last several years. The one that gets the most attention is, in fact, genome sequencing and RNA sequencing, nucleic acid analysis in general. And it now costs us about $850 to sequence a human genome, although the interpretation, at least the way we do it, is several $1,000s. So the bottleneck's not the sequencing, it's more the interpretation. And this number will drive down to a few $100s in the future. So all of you will have the option of getting your genome sequenced. The question isn't, can you get your genome sequenced? It's whether you want to get your genome sequenced and what you might learn, and that'll be an important theme for today's talk. And just out of curiosity, how many of you are interested in getting your genome sequenced? Wow, okay, how many don't want to get their genome sequenced? Yeah, only a few, yeah, okay, interesting. Yeah, there's no right or wrong to this, obviously. Anyway, you'll hear about my personal odyssey, so that can influence you possibly one way or the other. The other area that doesn't get quite as much attention, but is also, I would argue, a pretty big revolution going on is mass spectrometry, where we can now profile thousands of molecules in blood and urine and so bodily fluids that now make it possible to get biochemical assessments of folks. And certainly in many areas now, I would argue emerging technology are these wearable devices, and I'm going to talk about all of these different technologies as we go along today. So just by way of genome sequencing, I think most of you know this is having huge impact in medicine. This is just a few areas. And I'll spend a little bit of time on this, but then I do want to get into using other omics and big data to managing health. So basically, genome sequencing, again, you probably appreciate it's having an important role in understanding and treating disease. So especially in the area of cancer and solving mystery diseases. I'll spend a few more minutes on this. I won't have time, but you probably know genome sequencing is starting to have impact in prenatal diagnostics so that you can actually sequence fetal DNA from a pregnant woman's blood. So 3 to 10% of DNA in blood of a pregnant mom, depends where they are in pregnancy, is fetal DNA. And this is now being used as a standard test for trisomy of chromosome 21, 13, and 18. Although in principle, you can use it actually to get the whole genome sequence, which will probably happen in the future. An area that we'll spend most of the time on this is this one. What if you're healthy? Well, can genome sequencing be powerful? And so we'll come back to that. So just say a few words about cancer and solving mystery disease. I'm sure you appreciate that cancer is a genetic disease, both in terms of predisposition as well as changes that occur as people grow and become adults, and somatic changes. So it depends on the cancer, but you can have anywhere from up to, say, 10 or 20 different mutations in a genome that will make cells grow uncontrollably. Some cancers it's less, other cancers it's more, but typically it's multiple mutations that drive cells to grow out of control. And what we've learned now from something called the Cancer Genome Atlas and other large projects is that thousands of cancers have been sequenced of all different types. And if you've sequenced one cancer, you've sequenced one cancer. Every person's cancer is unique. The mutation spectrum there, it will differ. It is true that the mutations often fall into particular pathways, but they will vary again from person to person. We'll go through this in a minute. These are just some of the pathways from a paper from Bert Vogelstein. They're often mutated in many cancers. Again, typically it's certain pathways in some cancers, other pathways in others, but usually multiple pathways. And so the name of the game these days for cancer is that what's being done quite a bit is that one will sequence genomes of cancer normal tissue and try to find the genetic mutations that occur as cancer rises and identify those that are druggable. So just to walk you through this, these days this is very common. Again, in either blood cancers, case of solid tumor, you'll isolate DNA, sequence it typically fairly deep. We usually sequence a normal genome to 30 to 60x. So that means every base gets covered 30 times. And then that lets you deduce the variants. Here you sequence deeper. Just out of curiosity, why do you do that? This is for the undergrads. Why would you sequence cancer cells deeper? Yep.
Because they're going to have a higher level of variation, because each cancer cell is going to be mutating differently from the ones around it.
Yeah, there's a lot of heterogeneity there, yeah, and you want to capture that by sequence. So these days, some people say you should sequence 1,000x. It's also the case there'll be normal cells contaminating as well, so for both those reasons, great. So anyway, that's the name of the game. Sequence this, sequence that, try and find variants that do arise and see which ones are druggable. And so this is, in fact, one of the very first cancers we did. It was 2010. We had an individual that had metastatic colon cancer, so the cancer had spread, which is not a good situation to be in. When we sequence their DNA, in this case, a little bit complicated, but this refers to how many copies of chromosomal region they'll have. And this individual had two regions that were amplified, where there were 7 to 10 times more copies. And I see the label got lost, but this one has a gene called the EGF receptor, which is a particular called oncogene that when overexpressed can cause cancer. This actually had something called CDK6, which is also an oncogene, but was not as well studied. And what was powerful about this is that although this gene was known to be mutated in colon cancer, it was only about 2% of the cases. So by sequencing this individual, we discovered that, in fact, this person was amplified for that. Not only that, there were 2 drugs on the market, not for colon cancer, there were 2 drugs that targeted this gene, EGF receptor. They weren't there for colon cancer, they were there for lung cancer. And so what happened was after surgery, this person actually went on one of those drugs, again, a lung cancer drug that targets this gene, and this person's been cancer-free ever since. So, and the big point of this is we only discovered this because we sequenced their DNA. We would not have discovered it had we just run them through standard colon cancer treatment. Okay, so this is the power of precision medicine. That you can find changes that you can specifically target that are druggable. And for some cancers, this is absolutely essential. So lung cancer is probably the best example. There's typically 5 different pathways, and it varies from patient to patient which of these pathways will be mutated. So here's that EGF I mentioned before, but HER2, which is common to breast cancer. So any one of these pathways can be changed in lung cancer, and then depending which pathways change determines which drug you'll get. And if you have, say, a change in BRAF, and you don't get this drug, you get one of the others, it's worthless. It won't work. So the goal, again, is to match the medicines with the particular genetic defect. And so this is becoming very common in cancer. In some cases, like lung cancer, it's pretty essential, I would argue. The other area where genomics is having big impact is in solving undiagnosed or mystery diseases. So you may not realize, but 0.4% of children are born each year with a pretty serious birth defect. And parents, they'll start looking for what the underlying problem might be. Half the time it gets figured out right away, and the other half the time it's not clear. And so parents go on these odysseys where they try to actually figure out what's wrong with their child. And these days, it's pretty common to turn to genome sequencing to try and solve that case. And so it turns out, and in fact, actually by adulthood, it's realized that actually many more things. A lot of cardiovascular defects don't show up till adolescents or young adults because that's when the heart enlarges. So the number of people with underlying genetic defects is actually fairly large. But anyway, people do turn to genome sequencing, especially for kids, where it's, if you see a newborn child with a serious problem, there's a good chance it's genetic, not necessarily epigenetic. And so people are turning sequencing. This was first done, in this case, this is not by us, by Howard Jacobs' lab. This is a case of Nicholas Volker, who was normal up until age 2, but then actually started having some issues. First cut that didn't heal, then a lot of sepsis, a lot of problems in his intestine. He ultimately had over 100 surgeries, had his entire colon removed, and his health was declining very, very rapidly. And so they turned to genome sequencing and actually found a mutation in this gene, which is an immune gene, which actually suggests, well, he needs an immune transplant. So they did a blood cord transplant into this child, and there he is. He's been healthy ever since. So he actually got a new immune system, and it turns out in hindsight, your immune system is always in play, if you will, with your gut, because that's where you have more immune cells in your gut than anywhere else, because that's where microbes are, and they're playing with each other, so to speak. So this basically solved this case. Now, this works about 30% of the time. This is a more typical example where we did this case. We sequenced this child who had developmental delays, intellectual disability, series of issues, liver problems going on. We sequence this. When you do this, we sequence the child, the father and mother. When you do this, you'll see thousands of mutations in the child that they're either recessive in the mother and father, or some of them are de novo. But the point out of this is when we sequenced this, we had 8 candidates, any one of which there are mutations in each of these 8 genes. Any one of these could have been responsible for this situation. Okay? So we were kind of stuck. The way this one got solved is a second child got sequenced at Duke, and they posted the information on a blog of all things. The child had very similar phenotypes to this one, very similar situation. And one of those mutations was NGLI1, and the match got made that this child had this problem. One of the changes was in the gene, it's recessive mutation 2 change, which matched this one over here. And because these are rare disease, it's pretty clear this is responsible. There are now 38 kids with this disease that have been identified worldwide that have mutations in NGLI1. They all have fairly similar phenotypes. So this one got solved, if you will, by social media of all things. And if you think about it, this is a crazy way to be solving diseases. So there are now programs set up called Matchmaker, where you can try and share your variants to try and make these sort of matches through some of these genome centers. There's variations of this, but that's the general idea, where if you sequence this, you can run these 8 variants up through this and see who else has a similar case, whether that case phenotypically matches the case you have. So they're trying to use crowdsourcing, if you will, because recurrence, finding the same mutation twice, is probably still one of the best ways to figure out whether you have the disease. There are other things you can do too. You can do biochemistry and such to sort this out as well. So that's the mystery disease. The lessons learned out of solving these is, again, about 30% of these cases do get solved. In our case, we have yet to find any that's directly actionable on the affected child. But it is true, parents are still very grateful to learn what's wrong with their child, because they're typically searching often all around the world, to try and understand what's wrong with their child, and this does solve the disease. In some cases, parents want to have a second child, so they turn to vitro fertilization and then select embryos that actually lack these mutations. So that's how things are moving forward in the future. So, okay, so that's what I wanted to say about genomics and disease. And now, well, we'll save questions for the end. Now I want to talk about this area. What if you're healthy? Is genome sequencing valuable to you? At the time we started this project, most people said genome sequencing is a terrible thing to do. That what you're going to do, you're going to sequence people's DNA, they're going to see these genetic changes, we won't know how to interpret them, and if we do, you're going to scare the bejabbers out of everyone. And so the net result is you're going to turn everybody into hypochondriacs. It's going to cost millions of dollars per person. So the whole thing is really a bad idea. And I can tell you, I got that directly stated to me many, many times. Well, we wanted to actually test that, but I forgot to say, so the idea is that genome sequencing might be able to help you predict risk for disease, catch disease early, monitor and treat disease as well. And you would use other sorts of information, family history, what you know about medical information to help make to help do this in the most informative fashion. So as I say, most people are kind of scared of this. We decided we would go ahead and test. But we actually wanted to take it one step further, just thinking that genome is only part of what's going on. So we set up something we call personal omics profiling, where the idea is not only to sequence people's genome, but now we measure 14 different omes, if you will. We measure their epigenome, which for us is the DNA methylome, the transcriptome, all their RNAs, their proteins. We use something called TMT labeling and mass spectrometry, which lets us follow now about 7,000 proteins in peripheral blood cells. We have high throughput assays for cytokines, which are important, immune molecules. We've spent now about 4.5 years optimizing metabolomics and lipidomics so we can profile about 1,000 known metabolites and lipids and tons of other peaks, but we don't know what they are. Using mass spectrometry. So we do that out of blood and urine. I forgot to say most of this is out of blood. Autoantibodies, we've collected samples for 5 different microbiomes, but we've only analyzed 2 so far. And then about 4 years ago, we added biosensors, the wearables, I'll tell you a little bit later. And literally every time we sample someone, we're making billions of measurements. Okay? So we follow a regime shown here. Basically, we sample them about every 3 months when they're healthy, and then when something evil happens, like a viral infection or something like that, we'll take more, as you'll see in a minute. So why are we doing all this? Well, if you think about medicine the way it's done now, it's very focused on disease. So when you go to a doctor, you're usually sick, and then they try and figure out what's wrong and solve you, if you will. What we're trying to do is understand what it means to be healthy and what it means to be healthy at an individual level. So how similar are people in their biochemical profiles and how do they differ? We're trying to understand how different omes relate to one another. So I'm a believer that if you know someone's genome, epigenome, immunome, microbiome, by knowing what food they eat, you should be able to make a prediction about their metabolome. Knowing all the inputs into a system, you should be able to calculate the outputs, no matter how complicated it is, because these things are, even with big data, they are solvable. We'd like to understand how people have similar and personal responses when faced with a perturbation. So if I threw a rhinovirus on this room, some of you would get, maybe, depends on the dose, of course, maybe all of you would get sick, or maybe most of you would get sick, some not sick, some would get, probably most would get congested, but some might not, some would get headaches, others don't. So we have common symptoms, and then we have personal ones, and we'd like to understand what that means. And then lastly, when you do this very, very deep profiling, we will find things that do affect the health of the individual. So as you may have heard from some other things, and Steve said some of you have covered this paper already, we actually I was the guinea pig to get this going, and I didn't know I'd be interesting. It was really just to get the technology going. But it turned, so we set up this profiling on me, and yeah, basically the scheme is what I said. We've been doing it now. March will be 8 years. I've given over 250 samples. I've been through 11 viral infections, Lyme disease, a bike accident where I woke up in the hospital, And I don't look for these things, mind you, but if they come along, I might as well get good data out of it. That's how I view this whole thing. So anyway, we sample people every few months, and then when a viral infection or other things happen, we'll take many more samples. So 9 months in the study, got this nasty respiratory, these are all common colds. This is just the first few years, say 3.5 years of the study. But after, these are all common colds. This is respiratory syncytial virus. So each number here is a day. So as soon as I get congested, I'll take blood. You can see the next day, 2 days, 2 days, 3 days, so on. So dense profiling when ill, and then every 2 to 3 months when healthy. Interesting times like before and after China, things like that. So see what's going on. Okay, so we started with my genome, which made many predictions. I'll come back to this a little more later for other folks, but this is my complex disease risk profile. So for hundreds of diseases, you can sum over the risk alleles relative to the protective alleles for any given disease. So for example, high cholesterol sort of shows up here, I guess, high triglycerides and lipids. Maybe coronary artery disease is a better one. So there's a number of loci that will contribute to this. You can either have, again, risk alleles or protective alleles, and you can sum up over these. This is something Atul Butte set up. And so for hundreds of diseases, 100% chance of getting this disease in my lifetime. This is just the top of my profile. Here's a 0%. It's geared towards a European, you gear it towards your ethnic group and your sex. So this one's geared towards a European male. And that triangle is my starting point. And then the genome predicts either increased risk, that's the orange line to the right, decreased risk, blue line to the left. And my riskogram, if you will, pretty much matched what I knew already. That is to say, no one in my family, going back to my great-grandparents, has ever been overweight, so I'm very low risk for obesity. I have high cholesterol, such shows up here, high lipids, high triglycerides, and that actually runs in my family. I take statins, I'm a great responder to that. Coronary artery disease, everyone on my father's side has died of some sort of heart issue. Often it's thought to be coronary artery disease. He had 9 brothers and sisters, all but one died of a heart problem. And then 2 surprises. One came up late in the study, but high risk for basal cell carcinoma. And so I have 2 brothers, 3 sisters. They all came to visit me, and naturally we took blood from all of them so we could profile them just like me. And I just happened to mention the 2 brothers and 1 sister, what do you know about basal cell carcinoma? And it turns out each of them had basal cell carcinoma. I just didn't know it. So what I like to say is your genome is a better record of your family history than your family is, because your information transfer is better. You don't talk about basal cell carcinomas around the dinner table, or at least we don't in my family. And so from the genome we could discover this, but not from, again, transfer. And early in the study it was discovered as high risk for type 2 diabetes. That was a surprise to me. And because we were doing all this detailed profiling, one of the things we were following was glucose. And amazingly, when I went in for an insulin resistance test, was here, they measured my fasting glucose, and it came out high. I was very surprised. Measured again a week later, even higher. And this is a better measure for steady state glucose. You may know hemoglobin A1C. And it's arbitrary, but diabetics classified as 6.5. And I came out at 6.4, so I'm right on the edge. And 5 weeks later, I went to my general practitioner, took samples again, even higher glucose, 6.7, classified as diabetic. That was April 11, 2011. Why do I know that? Because up until then, I had the world's worst diet. I ate lots of desserts and sugary drinks and all that stuff because I didn't know I had a problem. And I cut all that out, and then I had been biking. I doubled my biking, started running, and that's about 8 months. Took a long time, and I brought it down to control. So this made a big splash at the time because the first time someone used their genome to predict their risk for a disease, in my case, managed it. And it's also interesting because of when it occurred. You may notice it occurred right after this nasty respiratory syncytial virus infection. So the hypothesis is that I'm genetically predisposed for type 2 diabetes. It is type 2. And then in conjunction with this environmental factor, RSV infections, what triggered the disease. That's the hypothesis, something we're testing with the larger group. So some people say, well, Mike, you don't know, that might have gone away on its own. So we accidentally did the right experiment because you'll see in a minute, we scaled now, we're studying over 100 people. And as we were doing this and everything is marching along fine, we were scaling this, collecting all these samples, a big project. And nobody's really looking at the data, including me, so we're collecting all this data, and if you will, I was blinded, and then right here, somebody looked at it and said, Mike, do you realize your hemoglobin A1C is 7.0, you're diabetic again? So no, I had no idea. I sampled me a few times, started running, I got it down, but the point is, there's no question I have a glucose misregulation problem. And it turns out my brothers and sisters are all elevated hemoglobin A1C too. They have the same problem. In fact, it turns out there's a lot of folks who are pre-diabetic running around. I guarantee there's a lot sitting in this audience who have no idea that you actually have glucose misregulation. But anyway, not to scare you, but we'll come back to that maybe later. But anyway, the point out of all this is, again, I do have a problem here. I did get mine under control, but that actually was from 2 years ago. And to be honest, it's been creeping up ever since. And you can't tell, but I switched from running to actually weightlifting. Like I say, you can't really tell. The logic was that increased muscle mass is better for glucose homeostasis. That actually failed miserably. I'm actually 6.8 on my hemoglobin A1C. It's just measured. And so the point is that, anyway, I, again, have a problem. I'm actually about to go on metformin. I got a flu vaccine. I wanted to wait until that. I sampled through that, and then I'll do the metformin. So anyway, again, I clearly have a glucose misregulation problem. I may have to skip over this, but and just tell you the result. But we've also looked at my epigenome, which is, we think, just as important as your genome. What's kind of, you may know that your epigenome is super cool because you can modify it, but not clear how yet with nutrition and exercise. And my favorite study is out there. There's a group in Sweden that had 23 people biking with one leg but not the other for 3 months, 4 times a week, 45 minutes of time. And then they sampled muscle biopsies from the biking leg and the relative to the non-biking leg and before and after. And there are 4,000 methylation changes that occur in the biking leg relative to the non-biking leg and about 2,000 gene expression differences. So if you think about it, so methylation, I forgot to say, it affects gene silencing. So think about it, that's exercise covalently affecting the covalent modification of your DNA and in fact gene silencing. Now, how that translates into health is not really clear. That's a problem you guys could actually solve in the future. I think it is a solvable problem. And so it would be cool to do that. Anyway, I'll just tell you that we looked at my methylome and my transcriptome. We haven't published this yet. My transcriptome bounces around like a yo-yo. Every time I get sick, it goes off. And that's no surprise. And that's shown here. But what was kind of cool is that my methylome over the first 3.5 years only changed twice in spite of all the viral infections. I have little kids, by the way. That's why I have, well, they're not so little anymore. But anyway, the point is that's why I was getting sick a lot. But it turns out my methylome only changed twice. And the 2 times it changed is when my glucose went off. So we think methylation is a measure of chronic disease. Your transcriptome is a measure of acute disease, if you will. And in some ways that makes a lot of sense because aging, your methylome changes with age, and that is a chronic condition. So anyway, that's a fun note. So as I mentioned before, we're now doing, to 105 people, what we've been doing to me. About half of them are pre-diabetic. We're basically sampling them every 3 months when they go through a viral infection or other stress. We take many more samples. We have all kinds of people doing cool dietary perturbations and trying to see the effect of these on people's profiles. And what's very cool about this study, I don't know who here is informatics, but nearly all of our participants have agreed to make their data open access, including their genome sequences. So you can get, it's a really rich source of data. We have over 1,000 time points, if you will, with these very deep data sets associated that are actually being made public that you will get access to. So just to run you through, as you might imagine, when you do a project like this, you learn lots and lots of cool stories. And I'll just give you a few vignettes here. So one criticism some people say, Mike, you learned something about your genome, but that was a fluke. How often does this really happen? So from the first 70 people whose genome we poured over just like mine, 12 of them have clinically actionable information. That is to say, they have mutations that with a reasonable frequency could lead to disease. Doesn't mean they will, but they'll get screened for that as a consequence. And perhaps the most famous, many of you have heard of BRCA mutations, right? BRCA mutations put women at high risk for breast and ovarian cancer. 50% of women will get one of those if they have a BRCA mutation. And so one of our individuals, it turns out, did have a BRCA mutation. And others have had other sorts of mutations as well. Many of them are associated with cancer. So all these will have a certain test. In 3 cases, they're associated with phenotypes. This one's interesting. This individual has a mutation that suggests he might have a heart issue. And then he did something called a stress echo, and sure enough, he actually has a heart defect. He's on drugs now. And he's a very young person. We actually discovered him. This all was discovered through sequencing his genome. So it's not great news to hear when you hear this, but on the other hand, it's pretty important for managing their health. This one's quite interesting, this MODY mutation. Where's that? Oh, up here. So this is an individual classified as type 2 diabetic, but there's a different kind of diabetes. It's called MODY, which is very single gene penetrant. And the way you treat a MODY person is 100% different from a type 2 diabetic. So in fact, once we saw this and realized they're a MODY person, they switch their medication. In fact, there's a famous story of a type 1 diabetic who late in life discovered he had a MODY mutation, and he estimated he had 21,000 insulin injections over his lifetime. And had he actually been sequenced at the beginning and figured out it was a MODY, he would have needed a total of 0 because he would have been on a different medication and he wouldn't have lost his islets and then had to take all these insulin. So that's an argument for why you want the right understanding at the right time. So anyway, so we find useful information for all these folks. I can tell just by doing these deep profiling, I won't have lots of time. We also found another person with a heart defect as we were profiling. We caught one person who had an early lymphoma, another person with a precancer called MGUS. They have way too many IgM cells that are growing. It's benign now, but you would screen for it. And lots of other sorts of things. This is just some of the diabetic information about quite a few folks were pre-diabetic who had no idea. Many of them were insulin resistant. That's not normally screened for. This is a big deal because you want to get your glucose under control early before you've lost. If you wait too late, you'll start making too much glucose. Then your body responds by making more insulin. You actually start making antibodies to your islets, and you'll actually make yourself a type 1 diabetic, but starting out as a type 2. So you want to catch these glucose misregulation things early. All right, I want to tell you about the wearable stuff. I'll just summarize this part, but we have a pretty cool study where we look at the effects of weight gain, weight loss, doing these detailed profiles. And about half of our folks are insulin resistant, the other half are insulin, they're normal. They're typically overweight folks, and insulin resistance is sort of a precursor to type 2 diabetes. It means their cells aren't responding well to insulin. The upshot out of all this is that we can tell from profiles that insulin resistant from the sensitive folks, no matter what ohm we use. So they have very different signatures. But one of the cool things that happened was we had people, they would overeat for 30 days, an extra 1,000 calories a day. And then they would lose weight for the next 60 days, come back to baseline. So we sampled in the beginning, after they gain weight, after they lose. And 2 pathways were incredibly strong that activated when people gain weight. One is inflammation. So your inflammation pathway goes up when you gain weight, comes back down when you lose. And the other one was both the dilated and hypertrophic cardiomyopathy pathways as you change. So your heart pathways are actually shifting when you gain weight. And again, they're reversible when you lose. So we think this might provide a molecular explanation for why folks get heart attacks when they gain weight. This would be the possible molecular underpinnings of this. All right. The other thing I'll tell you, and I won't have time, but everybody's profile is different at baseline. That is to say, you're all different from one another in your cytokines and your metabolites, and some of these are very, very important molecules. All right, I want to spend the last time on this because I think it's kind of fun. So just out of curiosity, how many of you are wearing a wearable device? A few? Okay, yeah. So at the time we started this, there were about 500 on the market. Now I think there's about 1,000 health-related ones. So we evaluated many of them. We looked for ability to get data, which ones are most accurate, so on and so forth. So again, there's all kinds of stuff out there. In the end, we settled on one smartwatch and a free Moves app that goes on an iPhone for all of our participants. And then I actually use 8 or 9 of these devices every day. In fact, I'm wearing 4 smartwatches as we speak here, so you can see these things here. And you may have noticed this is air monitor, and I'm happy to say I have a radiation one. So they measure all kinds of things. They measure activity, heart rate, skin temperature, sleep. This is blood oxygen. That's a different monitor. Activity, as I say, radiation, and I'm happy to say Salt Lake is very low. I'll double check this room here while we speak. Let's see what that one looks like. Yeah, you're lower than Palo Alto. So anyway, okay. So anyway, they measure all kinds of things. What's powerful about these devices is that most of them are continuous measurements. You're making hundreds of thousands of measurements a day. So one of the smartwatches we have makes 8 million measurements a day, okay, of all sorts of things. And then the name of the game here is to try and define people's personal baseline and then find deviations from that baseline for interesting things. So this is just one example. This is my circadian pattern. This is me, a few months of data. Here's sleep, heart rate, skin temperature. So I sleep at night, awake during the day, occasional nap, back to sleep at night. Heart rate, low at night, high during the day, back down. Skin temperature, which is different from core temperature, high at night, low during the day, comes back up, so on and so forth. So you get people's baseline patterns. I can tell you activity patterns are also very personal too and go different amongst people. That was me. This is, in fact, 43 other people. We have now many more. Everybody has a different baseline resting heart rate, blood oxygen, heart, skin temperature, what have you. We're all different. Again, no surprises there. That's basically what this says. Oh, this is the activity pattern. We all have different activity patterns. These are people who are kind of monotonic during the day, so don't move at night, uniform during the day. These are morning people, morning, afternoon people, morning, afternoon, evening. So you can classify people. We use algorithms to do this systematically, so it's not just eyeballed, although the eyeball does work pretty good. All right, so again, we're finding people's baseline patterns. As I say, the next step is to try and find deviations from those patterns. And it turns out this is one of my biggest deviations. This is known already, but your blood oxygen goes down on airline flights. Okay, so this is a short flight, San Francisco, San Diego. That's altitude in green. Plane goes up, comes back down. Blood oxygen starts high, drops low, constant pressure, back up, so on. So that's me. What's not clear from the literature, how low does it go? How long is it low? You won't find that anywhere, at least I couldn't. And so it turns out, we now know there's an underestimate, but 70% of the flights, your blood oxygen will be, for me, 96 or below. For other people, it's different. And 5% of the flight will be 90 or below, which is getting down there a bit, okay? What also isn't so documented, well, what does that mean physiologically? Well, it turns out, oh, that was me. Here's 18 other people. Now we've done tons of people. With one exception, everybody's blood oxygen will drop on a flight that goes up a certain height. By the way, if you want to know the air pressure, you can get that on your smartphone too. You can get a free barometer app that you can download to measure air pressure on planes. Okay, so everybody drops their blood oxygen on airplanes. So what does that mean physiologically? Well, it turns out we found out it correlates with fatigue. So you can do a blinded test that, classify yourself as alert or tired. And this is on the ground, this is in the plane. For me, that magic number is 96, the one I told you before. So when it's 96 or below, I'm typically classified as tired, 96 or above, alert. And here's a reaction time test. You have fast reaction times when your blood oxygen is high, and it's slow when they're low. So we think the reason you're tired on airplanes isn't because you've been working too hard. It isn't because you've been staying up late partying. It's because you've been the air pressure's low on the plane and your blood oxygen's dropping. That's why you're tired. So if you're a workaholic like me, that's very disappointing news because it means, no matter how fired up you are, you get on a plane, you're going to conk out. So, but here's the good news. If the flight's long enough, you can adapt. And for me, that number is 7 hours. So here, Frankfurt to San Francisco, planes, blood oxygen's high, flight takes off, goes low, here's a nap, sometimes goes lower, not always, but comes back up. For me, again, 7 hours, that's one flight. With many flights, same thing. Okay? So what that means is if you have a lot of work to do, hang in there. If it's a long flight, you may still get it done, I guess. So that's all I'm using about plane flights and such. Well, how does that impact health? Well, again, when you're defining people's personal baselines, you're looking for deviations. And it turns out these wearables were invaluable for me for detecting when I had Lyme disease. So I don't know how much Lyme disease is in Utah, But the backstory is helping my brother put up fences in rural Massachusetts. And then 2 weeks later was flying to Norway through Frankfurt. And on that last flight, which is pretty short, it turns out my blood oxygen was way lower than normal. So because I measure myself all the time, including coming here, I knew what my blood oxygen should look like on a short flight. Drops down typically 95, 96, comes back up. This flight, the median was 90, and it never came back up. It stayed about 90, 95, 96. Not only that, it turns out my heart rate was high, and I later learned my skin temperature was high. And this is some of the data for this. Is skin temperature here, heart rate here. And then I got a low-grade fever, and I went to a doctor in Norway. I warned him it might be Lyme disease because of the timing. And he took my blood, my monocytes are up. He said, yep, you have a bacterial infection. You should take penicillin. And I said, no, I think I should take doxycycline. And as you might imagine, it was pretty tense for a few moments there because are there any MDs in the audience here? Yeah, they don't really like to be told from their patients what to do. So anyway, he reluctantly gave in. And then a day later cleared it up. Then when I got back, I was tested, and sure enough, I was positive for Lyme. I had seropositive for both the antibodies and antigen. I had the perfect control because I had given blood a few days earlier. It was negative, so it's not a false positive test. So very well controlled experiment, very clear. I had Lyme, and this was all figured out, again, from my wearables. So that prompted us to go back and look at the 2 years I was wearing a smartwatch and see how many times I deviated from baseline for heart rate and skin temperature. And it turns out there were 4 periods. One was the Lyme, which is, you can't miss, these are days by day resolutions. So the Lyme signals are so strong, these are outlying heart rates. And then it turns out there were 3 other times, 2 of which I knew I had a viral infection. The other I didn't classify myself, but when I looked at my CRP, it was very clear I had high CRP, just like a viral infection. I just wasn't reporting being symptomatic. So every time I was sick, either by knowing I was sick or probably not knowing I was sick, my heart rate was up and my skin temperature was off, okay? So that led us to write algorithms where we think we can tell, this is now over, we now do it over 30-minute windows, but over 2-hour windows, what this one said, where you look for differences in outlying heart rate measurements. It's better than skin temperature. You look for outlying heart rate measurements relative to your baseline, which tell you something might not be right. And with that, this was plotted as a delta plot. It's complicated. So for each of the periods I was sick, we could tell when my heart rate went up before I reported being symptomatic. We have 3 other people, one of whom got sick twice, and it works on them too. So that is to say, we think from a smartwatch, we can tell when you're getting sick before you know it. Okay, because your heart rate goes up, or it might be when you're first starting to feel a little funny. And so we think that will be very powerful so that, you don't necessarily go into work and infect everybody in the future if we can tell when you're getting sick. I don't have time for this, but we also think we can tell diabetics from pre-diabetics from pre-diabetics because they have different heart rate patterns as well. And in fact, there's a study that just came out I saw in the press, I haven't seen the paper yet, that claims they can do something similar with slightly different variables. So the point is, I think these wearables are going to be very, very powerful for detecting disease. And so in the future, I envision that your smartwatch is actually going to be your personal health dashboard, where all this information will go into your smartwatch, including ultimately and we're building this now to try and get your microbiome and all that stuff in there, so that this will become your personal dashboard, just like your car dashboard tells you when things aren't working right for your car. And it shouldn't just work for you, it should also work for your physician, because there's no reason not to share all that data with your physician. I can tell right now the data you get in a doctor's office is garbage compared to what you can get off these wearables. Most doctors, the first reaction is, well, your wearable data is no good. That's actually completely false, because when you go into the doctor's office, you're often rushed or whatever, and your heart rate's all over the place. Mine varies by 30 BPMs. Okay, and I don't get nervous in the doctor, but it depends whether I rode my bike or I have a grant through that day or whatever. It's all over the map. But with the wearable, my resting heart rate's the same every day, unless something's off. Okay, so we think this will be very, very powerful. So next time a physician says, well, when did you first report getting sick? You don't have to scratch your head. You just look at the heart rate and say, well, it was 3 weeks ago. It'd be very obvious. So we think this will be the future. And so this is Mike Snyder's world. I envision a future where people will be getting their genome sequenced before they're born with other kinds of omics and wearable measurements, And with that, we'll be able to better manage folks' health. I think I want to leave time for questions, but the bottom line out of all this is that we think some of these big data technologies, they are here, they're starting to get used. I think the most important part of all this is the fact that these are longitudinal studies. We're trying to find people's healthy baseline and deviations from that, whether it's a wearable, whether it's an omics profile. And that's what we think is going to be important for managing health, rather than trying to figure out people when they get sick. That's very hard to do. So we think that understanding your deviation from your personal healthy baseline is what precision medicine is and should be all about. And I also think people are going to have to be responsible for their own health, because no doctor in the future is going to understand you like you can understand you, I would say. Okay. So I'm lucky I've had an amazingly talented lab who's worked with me and collaborators. So Tracy McLaughlin's collaborator, George Weinstock on the microbiome. We are funded by the Human Microbiome Project, which just ended recently. But these are some of the key postdocs. Wenyu, Reza, Kevin, Monica, and others have been absolutely instrumental in making all this happen. And here's a shameless plug for a book we had come out recently. So if you want to learn more about some of at least the earlier stuff, this is some of it's featured in here. So I think I say some time for questions if you have any. So thanks again for having me here. Yeah.
I was curious about the methylome and bisulfite sequencing.
Yeah.
How does that work?
You mean how do you map methylation?
Yeah.
Yeah, sorry I glossed over that. But basically, when you have methyl Cs, if you treat DNA with bisulfite, all your Cs will convert to something like essentially U's that will get converted to T's when you PCR them. But it turns out the methylated Cs don't convert. So what happens is if you know someone's genome sequence, you treat with bisulfite, you either make a library and sequence all that, and wherever it was methylated will show up as a C, and wherever it wasn't methylated will be converted to a T. And then you actually, because methylation is often partial, you're counting the fraction of those C's and T's, if you will. Does that make sense? Yeah. Sorry to go through the method, but that's how you figure it out. It's very expensive, by the way, to do it for the whole genome the way we did. That's a several $100,000 experiment, actually.
So just on that same topic, PacBio says that they can actually detect the methylation now. Has your lab done any of the?
Yeah, we've talked to them about that. They're still even more expensive because they have long reads and you need to sequence them many times to capture that. And every time we talk to them, they always say, well, we're not quite ready yet. So it gives a different pause signal for the polymerase. And so I think in the future that'd be phenomenal, especially for the RNA. There's modified RNA to your epitranscriptome, and we have not yet explored that. Okay. Yeah.
Maybe we'll go from a zoomed-in question to a zoomed-out question. If you could give us kind of an Elon Musk vision of infrastructure for personalized medicine in a decade or two, how do you see? I don't think we'll be wearing 6 watches up our arm.
Yeah, great question.
In terms of data acquisition and data interpretation, right?
Yeah.
Where are the computations happening?
Yeah, no, that's a great question. So I envision the future, you'll need one, possibly a second sensor system. Could be a smartwatch. The problem with the smartwatch is it doesn't do SpO2, which we think, the reason we're exploring this is trying to figure out what measurements are most important. There's a few others that we're exploring now we think are going to be also equally valuable. SpO2 doesn't measure very well off a smartwatch. It will measure. It'd probably measure better off a ring or something like that, where just like, because you may know now you do a finger device for that. So I think we'll get by with 1 or 2. All that information will relay to your smartphone. And I believe it'll be just like a car. There'll be a whole back system of data. And of course, we're getting better and better at storing data. That's getting cheaper. And algorithms to interpret it. But I think just like your car, you may not know it, but your average car has about 400 sensors on it. And then there's just a limited number of lights that show up on the dashboard when something's not quite right. I think that's what your health system's going to be in the future, that this will all be running in the background and some supercomputer there. And that when, a blip goes off, that will show up, In fact, that's what we're trying to do now for infectious disease. We want to write a system where yellow light goes off when we think there's a a deviation in your heart rate that's abnormal, and if you're watching a scary movie, you just ignore it. But if you're, you're sitting around reading Time magazine or whatever you like to read, and suddenly your heart rate's going off, then something, unless you're reading something, I guess, terrible in Time magazine, maybe that was a bad one, but you get the idea that if you're in an unusual time when nothing should be happening and something's happening, there's something going on with your health.
That's the wearable side, and then there's the whole, all these other measurements, right? All the other omics measurements.
That's a great question too, yeah. Yeah, I think that'll be a finger prick of the future. What we're doing now is a research study. We make billions of measurements, like I said, but all of those aren't going to be clinically relevant. The problem is we don't know which ones are and which ones aren't. We're still sorting that out. We know from a transcript profile, we found someone whose liver function is definitely off, although we can't find out why it's off. Very interesting. But we know they're an outlier for their liver stuff. So that may turn out to be a signature, but we want to find the key molecules in that signature that tell that liver is off, and that's what we'll have to sort out. So it'll be research for a while. We just need millions of these data sets, and then I think we can find a few molecules that hopefully in the future, now when you go get tested, you get tested for about a dozen, maybe maximum 16 things. In the future, it'll probably be a few thousand things, but you should be able to do it all from a drop of blood. And in my world, I think healthcare would be like Amazon. If you think about it, the way we do things now, it's terrible. You go to a doctor, you wait forever, you get sick because everybody else around there is infecting you, then you go back home and you infect your family. So it's just not the way we should be doing medicine. We should be doing it all. You should go to Safeway I don't know what supermarkets you have here, but you should just put your finger on a little thing and out should come a report that says, oh, your CRP's up. Get the hell out of the store or something. This is how it should be in the future, that we should be, and there's no reason not to. These tests are not that expensive. Problem is we're in the learning phase. Yeah. And it's hard to change medicine. It's a very regulated field, as you probably figured out. Yeah.
Just a quick question. As you think forward to the future, are current laws for privacy and those things, are they going to change in mind?
So yes and no. You are protected for health insurance and employment, you may know, from the GENE Act. But you're not for long-term disability and life insurance. And my own view is that, and this should resonate well here, is that any wealthy country, which I would argue the United States is, should be able to take care of its citizens at some level. To not protect them for long-term disability, it just seems like a crime to me that we should have some safeguards for all this. Now, does everybody deserve deluxe care? I don't know. There has to be a practical limit to this. But anyway, so I would argue we need more. We have some in place. Yeah. Life insurance is arguable, I would say. Steve?
So 23andMe versus exome versus whole genome. Which, obviously you guys are doing the whole genome. You get all the regulatory studies and everything else. Does that just give you a ton more data than the
Yeah, great question. The problem with the whole genome is we don't really know how to interpret from a medicine standpoint outside of the coding regions. A little bit on the GWAS variants, which we used for mine. So these days, the best buy would be the exome, although pretty soon the prices will be pretty similar anyway. 23andMe only measures your complex disease profile. They have 2 predictive alleles, one for Alzheimer's, one for Parkinson's, but the rest are all just complex disease. And after the fact, we did 23andMe on me, and 70% match up with my riskogram, but they did miss the type 2 diabetes. But we had a lot more markers than they did for that. We had Atul Butte's an expert. Type 2 diabetes. So we had a bit of an edge there. So the bottom line is, though, you do need the sequencing, because all the rare variant stuff, which is highly penetrant, actionable information, like those 12 out of 70. I forgot to say that's only a minority of people, but if you're one of those 12, it's a big deal. And it's good to have that information. And that's what you want to capture from a sequence. And if you do have one of these rare alleles, you just get screened for it. And even if you're scared of getting your genome information back, which I can understand. Like my wife doesn't want to get her genome done. That's cool. I would argue she should get it done, but all the information should come back to her, because you don't want to wake up every morning worrying, oh, I'm going to get a heart attack. I'm going to get a heart attack. What you want is, but you do want to learn if you have a BRCA mutation, so that your physician can then screen you once a year, what have you. So, yeah.
Most of us won't, probably won't be found wearing multiple devices. If you were to choose one device to give you the most information, would you tell us what it might be?
Well, I would have said, and I have no stock in this, but Base's smartwatch. But the problem is they got pulled from the market because 0.2% of the devices overheated. So people got blisters, and they're bought by Intel, and it's not Intel's core business, so it's just easier to vanish it. But it was great because it's very high resolution, and it measured lots of things we're interested in. So then we're in different zones here. So the Sympatica watch is the best one, but very high resolution, but $1,000. And it has some non-friendly parts to it. So it depends on your budget and things. So the two most common devices are the Apple Watch and the Fitbit. And the Fitbit has certain things that I think will be powerful for the future, but they're in competition. So we'll see how that arms race, literally arms race, I guess, plays out. So I have, you can see I have one of each. This is the Ionic Fitbit that will measure some interesting things in the future. And the Apple Watch is somewhat limited. I do think the sleep information is useful. So for the moment, if I had to pick, I'd lean towards the Fitbit, but it's hard to share in a research study for reasons that are technical. Yeah. Okay. Good, thanks again.